Today's topic is method in gene cloning. The first step is isolation. We have to isolate the DNA vector from bacteria cells, and then we have to isolate the DNA that contain gene of interest. To isolate the vector, we must have a marker genes, either AMPR and LAZEC, and then we must have a restriction site. Then we can isolate the vector. Step 2 is cleave or cut. To cut the plasmid and donor DNA, we have to use the same restriction enzyme. If we not use the same restriction enzyme, this method will not work. When we cut the when the restriction enzyme cut the plasmid, they will make a single cut within the LAZEC gene. Next is insertion. When we then cut the plasmid and donor DNA, we will combine them together, and then we have to add the DNA ligase to seal them together by forming a phosphosidal bond. The product will include recombinant DNA and many non-recombinant DNA. Step 4 is transformation. As you can see at this paper, they have a 4 plasmid. It is because when we cut the target DNA, we will not get only target DNA. We also get gene that we will not use in this method. So that will that's why we got these 4 plasmid. The first one is what we want, recombinant plasmid. Recombinant plasmid is that have a target DNA, and non-recombinant plasmid that without the target DNA. Then normal plasmid, and then no plasmid. This plasmid will transform in the host cells, and then we will become a bacteria cell. The last step is screening, where we use a blue-white screening to screening the recombinant plasmid and non-recombinant plasmid. For recombinant plasmid, their LAZEC is distributed that cannot encode beta-galactosidase, so they cannot hydrolyze at scale. The colonized will be white. For non-recombinant plasmid, the colonized will be blue because contain LAZEC that encodes beta-galactoside, so they can hydrolyze at scale. So we will look at the white because they contain the recombinant plasmid. Thank you.